them. We will cover questions at the end of the two talks at the end of each of the prizes. Okay. So Hopfield, John Hopfield is really responsible for what we know as the Hopfield networks, and that's what I want to cover today. John Hopfield really so he received a Nobel Prize in physics. I've seen a lot of discussion in the web. Why physics? Well, to me, it's very clear that this is physics inspired. John Hopfield grew up as a physicist. Did his PhD in Chicago in physics. He then went to University of California, to Princeton, to Caltech, where he did the work that we're going to cover today. And went back to Princeton before retirement. I was actually there and had the fortune to meet him there at 1999. Key contributions, of course, a Hopfield network we will cover, but I would say almost like not less importantly, other contribution, like most notably kinetic proofreading, the idea of using how biology is using energy to decrease error rate in specificity. I'm noting it here because it's again this idea of getting ideas across disciplines from physics to biology and back. Okay, Hopfield network is really, it's based on, it was seeded in a paper in 1982, PNAS paper, authored by John Hopfield alone. I want to say I went now and reread it. Very simple paper. I recommend reading. In a way, there are many things missing in that paper, in some sense, but it was a major seed to really attract talent and ideas and revive the field. If you look, it is really, okay, so what it does, it suggests the concept of associative memory or content addressable memory, the idea that memory may be stored in ways that we don't reach it by an address in the computer, but rather by giving something similar in a similar pattern and retrieving it. It is inspired by biology, we will see that. And it really already put to terms the key wording that we're using today of generalization, pattern recognition, error correction, the idea that the network is not sensitive to individual neurons, but rather the idea of a collective phenomena. Okay, associative memory is the idea that we start with something similar. We have a set of memories. We give the network something similar to one of the memory, and the network need to reconstitute one of the closest memory. Okay, and we will talk about how that works. I want to say there are two prizes this year for AI that have been seeded, like this one early on in the 1980s. But these were surely not the beginning of AI. There was a big wave of very important ideas that were seeded in the 1960s, much earlier on, most notably by Frank Rosenblatt and others, doing fascinating things on even layers of network with layers and classification and such. But that has started to decay. The enthusiasm started to decay with what is known as the winter of AI. And in a way, the paper of Hopfield kind of revived the interest in the field. But if you look at the paper, it is actually not inspired by this work. It is inspired by much earlier work in physics from the 1920s of what is known as the Ising model. And I want to show you that. The Ising model is essentially a network collection of spins, electron spins, that try to align to their neighbors. And that really creates dynamic that lead to patches like that of co-aligned spins. And I think in Hopfield mind, he kind of looked at that and said, can I use that idea, but to create a pattern that I want? And the idea was very, in a way now is very simple. It is, again, inspired. This is the first figure from the paper, inspired by a neuron that has a kind of an action potential and a firing rate and a threshold. It says, let's approximate that by a simple square function. Here's my neuron, and my neuron gonna start firing if the input it gets from neighboring neurons that are also firing is larger than some threshold. Very, very simple. Okay? All neurons are connected, they have weights, and each neuron just sum all of the contribution from all other neurons that fire, and say it's larger than a value, I'm going to start firing. Okay. So now if we want to embed a pattern, let's say JA, John Hopfield, in a network, the idea is we're going to use what is known as the Hebbian, Donald Hebb learning. The idea that if two neurons are co-firing, the synapse should strengthen. If they're kind of not correlated in the firing, the synapse should weaken. So we go over all the memories we want to embed, and we use this condition to set the weights. And essentially, this is it. We now have a network, and if we give it a pattern that is similar to one of the memories, and I play it, it will kind of restore the memory that it had. If we give it something similar to another memory, it will restore the other memory. Again, with the inspiration of physics, and not just inspiration, the kind of the point of view, the foundations of physics, what John Hopfield did is showing that this network actually obeys an energy function, a very simple energy function. Let's go over all the pairs of neurons if they're and count their contribution with the weights of the interaction matrix, and that if you define that energy function, then by definition, mathematically, the energy always goes down. So you kind of have a fitness landscape on which the network goes, and that actually guaranteed that it doesn't go in cycle, that it must converge to a stable solution. Okay, how many stable solutions can you embed? That really brings us to the last figure in this paper. Again, it's a very simple paper, very, in a way, after introducing those equations that I've shown here, this is equation 8 in the paper, this is essentially it. It's just one simulation shown here, not very comprehensive in any way, not much mathematical foundation for what we see, for example, in this figure, but in a way that has been the seed to a lot to come. What we see here is a network with 100 neurons, and what John did is trying to teach it different number of memories. So first 5 memories, 10 memories, 15 memories, and he shows if you teach it 5 memories and you start with some random pattern, it would converge to one of the 5 without any errors. If you do 10, it would often converge to one of the 5 without an error, but sometimes there are errors. If you try to condense too many memories, it breaks. Quite often it doesn't breach any of the pattern that you've intended to. And John actually explains it in the discussion as the idea that from the energy point of view, that you kind of have minima like that. You want them to be separate, but when you start to have too many memories, they start to get too close, and what you get are new minima that you didn't quite intend for. These have been later on called spurious minima. You can kind of think about them as the modern day, what we call hallucinations in AI, something that the network invented that has never been actually taught. There's been a lot of work in trying to think how to increase the memory. One of them is by doing unlearning, by kind of not just learning these points, but also de-learning these wrong minima. That has really exploded in many ways. For example, just to mention one, the idea of actually promoted by Crick from Watson and Crick from the DNA, that maybe the reason our brain dream at night is because we kind of set it in a random place, it goes and find those wrong minima, and then we need to unlearn to forget those minima. But not just, I don't want to specifically advocate for this idea, but just to say how inspiring this whole work has been. Okay, conclusion. Hopfield network really introduced an idea of associative memory, the fact that we address memory not by, we retrieve memory not by an address, but rather by showing something similar and getting a pattern. It is inspired by physics idea, going back to Ising model, the idea of an energy function, and by biology, the neuronal activation and the idea of Hebbian learning, co-firing neuron tend to strengthen the synapse. And I want to say, in a way, the impact has been, a lot of it has been in kind of attracting talent from physics, from other disciplines to really rejuvenate the field of AI that have kind of started to go into this AI winter. As mentioned, it will take another 40 years from there to the huge explosion in AI that we see today. Okay, so I can end, and Dvir, it's you.
Okay, so let me move this. So I will continue, I'll move forward 40 years to Geoffrey Hinton on the birth of deep learning. So let's start by talking about Geoffrey Hinton. He's now 76, going to be 77 soon. He's originally from the UK, where he did his undergrad and graduate studies. I think his graduate was in Scotland, in Edinburgh. And then he moved to Carnegie Mellon, where most of the work in his 80s that he was awarded the Nobel Prize for was there. But then he moved to University of Toronto, where the paper I will be talking about came from. You see the screen I put here, Mount Everest, because apparently the E in Geoffrey Hinton's name is Everest, because his great uncle was George Everest, and the family, I guess, is very excited about that. And you can try to make how he reached to the top. Okay, so this is Geoffrey Hinton's Google Scholar page, where you can see only 860,000 citations, which put him, I think, third or fourth in the world today in Google Scholar citations. And he had many contributions. I think Hopfield had contributions until the 80s. Geoffrey Hinton started in the 80s, and his contribution since then, every year a paper with thousands of citations. In the Nobel Prize, their press release, they mentioned this paper here. You see a learning algorithm for Boltzmann machines. This is not one of his important contributions to the field of AI, at least in my opinion. I'll talk about very briefly. I think they wanted that because of the controversy that they knew that will happen of giving Geoffrey Hinton a Nobel Prize for physics, because unlike Hopfield, he never studied physics. He studied experimental psychology as an undergraduate, and then moved to AI, and all his research was not physics. And they thought that Boltzmann machine comes from physics, so let's talk about that. But his major contribution, and the paper I will talk about, is this paper here that is becoming probably soon going to be the most cited paper of all time, only 12 years ago. And it's a very important paper, and I'll try to explain why. And it's clearly the reason why, from all the people that contributed to this field, Shai Benjo and Jan LeCun and others, he was chosen to represent AI. Okay, so this paper, ImageNet classification with deep convolutional neural networks, that was published in NeurIPS in 2012. You can see the name of the authors here. Alex Vyshevsky is not very known today, although this paper is called AlexNet, because that's the method that was developed here. But Ilya Zatskover, we all know today, developed ChatGPT, and also Israeli, and Geoffrey Hinton. And it's very clear that this is one of the seminal papers. You don't see here Boltzmann machines. You see the Perceptron MLP, and then LeNet, which is in 98, and we'll talk about it briefly, and then comes AlexNet, and from there we move to ChatGPT. Okay, so what was so important about this paper in 2012? Because there's not a lot of novelty in anything that they did there. There's a little bit, but what's novel here is that they connected three things. The first ones to connect those three things: the algorithm with the hardware, with the data, and these three things together are what makes deep learning what it is. And I'll talk about each one of them separately, and I hope we'll understand why this is so important. So the algorithm that they used there was convolutional neural network, CNN, which they didn't invent. This is Jan LeCun invent, which is an algorithm that is based on neural networks to label images, and there is a part here of the convolutional part where you take, instead of trying to extract feature of the image, you take regions, pixels, and then you do all those convolutions and put together those from many images, and then you put it into a neural network. And this is what we're going to talk about, because although there is, you see the ReLU here is a contribution in this paper, this is not the main contribution. So Real already talked about neural network. I'll just talk about very briefly. So the idea is very simple. You have inputs coming into this node that is called a neuron that does a very simple calculation, taking from the inputs and the weights and the edges, and return 0 or 1. That's the idea of neural network, and that's not going to be very helpful to make predictions. But then we have this multi-layer neural network where we have many inputs, we have many neurons, and we can have many layers, and where this information passes through this network, we can apparently learn very interesting things. So we already talked about this Hopfield network, which was a very simple network to do this kind of learning. Hinton in this in the paper from 85 presented this idea of Boltzmann machine, and then a year or 2 years later, this restricted Boltzmann machine. Those are not used today. They were only a step on the way that inspired further work, but I'll talk about them very briefly. So the Boltzmann machine is you have the visible node, which will be your inputs, where you know what are their labels, and you have the hidden node, which represents some kind of features. We don't know what are those features, but they are the features. And you see here, everything is connected to everything in this network, and just like in Hoffield's network, the idea is to minimize the energy function, which in this case, and I think also in Hopfield networks, is this Boltzmann distribution or Boltzmann function, which comes from physics. So this is a way to connect this price to physics. The Boltzmann machine was never used because it's impractical to learn. You see the number of edges here, increase the nodes to, I don't know, 10 times more at least, and it's impractical. You have way too many edges. So then they have the restricted Boltzmann machine, where there are edges only between the visible and the hidden nodes, and no nodes between the nodes in each group. And I like this example that makes it very easy to think about it. So you have your inputs, which like a list of movies, whether you like them or not. And you have the hidden nodes, which you can think about them as features. Again, here there are names of features, but they are not really features. And while this flow of information goes through this network, then you can think about, you can have those specific hidden nodes that are firing, that are adding information to this node. So this person maybe likes Forrest Gump and Titanic, but not other movies. And then when you learn all those edges, then you can, and you try, while trying to minimize this Boltzmann distribution, then you can learn new information for nodes where you don't have information. So this is that. Geoffrey Hinton had a much more important contribution to the current state of AI today, only a year later in 1986, which is called backpropagation, which really moved this from Hopfield and Boltzmann machine, which were more pattern recognition, to this idea of supervised learning, where there is input and output, and the input, there is information flowing from the input through those hidden layers and going to the output. And then based on that and comparing the output to the known label, you can then go back and do this back propagation, going back in the network to adjust the weights and going back and forward until there is accuracy. Instead of trying to minimize the energy, now the measurement that you're trying to minimize is the accuracy. Okay, so this was, so in this paper, they presented this algorithm they called AlexNet, because Jan LeCun called his CNN LeNet. And what you should appreciate here, you see all the layers in this algorithm. And what is really important here is those yellows. So in the original CNN, you see three layers of three hidden layers. With 120, 84, and 10 connected neurons. But in AlexNet, they were able to increase that to 4,000, 4,000, 1,000, which brings you to this network has now 600 million parameters compared to, I don't remember the number here, but in the thousands. And how were they able to do that? And that's because of another revolution that started a few years earlier with GPUs. So CPUs, as we all know, are the processing unit that we use in our computer to do all the computations that the computer does, and it works in a serial manner. So you have your inputs. There is this in the CPU. It can do some function on it and return an output, and then that output can be used further on. In the GPU, there is this parallel approach where you can take this input and run thousands or millions of parallel tasks, exactly the same calculation that happens based on your inputs. And why is it important? And that was developed by NVIDIA in the 90s to do for video games, because in video games, you have the viewpoint, right, where you're looking at what's happening, and then you have the light can change. And if you need to now calculate each pixel every time you move your the light, then you'll need to recalculate all the pixels on the screen. That will take a lot of time. So the idea here is that you can calculate all the pixels together. And in the 2000 students in Harvard and started playing with this GPU and hacking it and using it for all sorts of other uses other than video games. And NVIDIA saw that and developed this package CUDA in 2006, which allowed users to use the GPU to do calculations for other things. And immediately it was understood that it could be used for AI. AlexNet was the first paper that used that for in the context of neural networks. And the data. So to do machine learning, we need data that is labeled, and here specifically we're talking about images to label images. And since the 90s, it was many groups started to create all those data sets with thousands of images with labels, and those were used as benchmarks for all the algorithms that were developed back then. But then Fei Li from Stanford said this is not enough. We need a much larger data set. And in 2006, they extracted images from Google, and then they gave them to mTurks to label them by hand, and they got to 14 million images in 20,000 categories or labels, and that was an enormous data set, which only got them a poster in the conference where they sent it to. But it took 2-3 more years, and it was understood that this is a really great data set. And a few years later, in 2010, they started this competition, the ImageNet Large-Scale Visual Recognition Challenge, where groups from all around the world could send their algorithm and try to predict a hidden set of images, the correct labels. So this works. They only took the top 1,000 labels with the most pictures, and your prediction should be in the top 5 labels. The correct label should be in your top 5 predictions. In 2010, 2011, where they used the traditional machine learning approaches of extracting features from the image, the classification error was 28, 26. And then other groups in 2012 also had 25 and 24. And I'm sure they were very excited about their chances of winning that year. But then AlexNet came with this idea of using neural networks on this data. And it was only possible because they were using GPUs. And it was only possible, and the neural net only worked because it had this massive data set. With smaller data sets, this wouldn't have worked. But this paper really made this result, really made everybody understand that this is where we need to go. This is much better than what we had before. And in the coming years, more layers, improve the algorithm, improve this until we got to a point where the algorithms are better than the human error. So just to summarize, this paper represented this combined together those ideas of neural networks with GPUs that allowed to create neural networks with enough neurons to really be able to learn something and connect it with big enough data sets. And since then, there are many very big data sets that have been developed, because this is the only way where you can really make those neural networks be more effective than traditional machine learning approaches. So I'll finish here, and I'll say that this brings us to Fabian's next talk and the next Nobel Prize, which I think Ray said how everything is interconnected, because AlphaFold really used the advances that were in this paper, in these works, to contribute to biology. So I'll stop here. And I think we have time for questions, right?
Okay. Yes, we are actually on time, which is nice. Thank you, Dvir. It has been really fascinating to hear. Maybe I think you went through that, but okay. First, I want to say, if people want to ask questions, please type them into the chat. I think that would be the easiest. Although, if you want to turn on your microphone and jump in, that should also be okay. I want to start maybe by asking you, Dvir, about the importance you see for back propagation, which was, I think you mentioned, but maybe very briefly, which is a 1980 something, 86 paper.
1986.
1986 is kind of a major seed for the later explosion in this paper and what we see today. If you can comment.
So I think it's, again, you showed this figure that I didn't put. of the AI major steps. You showed, right, the AI winter, and then the 80s, and then since 2012, we call the golden age of AI. It didn't have that much of an effect in the 80s. It was backpropagation because, again, the networks were too small to really make to be used. It only came back in the late 90s, again with Jan LeCoul and Yoshua Banjo that started using it in their more elaborate neural networks, which and again, not like Boltzmann machines, which I don't think you learn them in the university. Nobody knows about it. They're not really used. They had the inspired work, but nobody used them. Nobody uses them today. Backpropagation is really the main algorithm that is used to train those neural networks. And I think that it had more than that. It's not just the approach. And I think I mentioned that it's kind of the first time. I'm not even sure they understood it when they published the paper, but it really changed the use of neural networks from pattern recognition to moved it from pattern recognition to what we call today supervised learning, which it's not mentioned. I looked at the paper, I didn't see them mentioning this, but this really
Okay, I think
changes
I'm
the way
losing you, Dvir. I don't know
Right.
if it's on my side or on your side. Okay.
Can you hear me?
Oh, you're back. Okay.
I don't know. Sorry.
Okay.
But this is
I
really
think for me, just to kind of get it into the discussion phase, not that we're going to open it up, but I think it's really fascinating to think how the very basic ideas of how to do deep network were all there in the 1980s. And I think it still took 40 years to get to where we are. And it is, I mean, it's fascinating and puzzling and very interesting. And I think maybe...
But really, that's what makes Alex
trying to
Smith
push forward
so important, because neural network without the hardware that allows you to make it a big enough network and without the big enough data is useless. It doesn't work. It works worse than traditional machine learning methods. So the algorithmic ideas were there, but it was just not possible to make them work back then. So how do you say תקדים את זמנו, right? Those ideas, there are ideas that had to come from many different fields that all merged into this 2012 paper, which is clearly the reason why Hinton won the Nobel Prize, although they don't mention this as the reason, but this is his main contribution to the field. So that's why I think this paper is cited so much and is probably the most important paper of at least in the AI field, the most important paper.
Okay, there's a question in chat. I don't know if you know why it's called the Boltzmann machine.
Because they use the Boltzmann function there to minimize the energy of the network, which it's called the Boltzmann machine. But the funny thing is, as far as at least what I read, the Hopfield machine also uses the same energy function. So you can call Hopfield networks also Boltzmann machine, but he didn't use that name, and Hinton did use the name Boltzmann in his algorithm. So, but that's the reason. And then there's also something for you.
Okay.
Because of inspiration from Ising models and okay. So I don't think the main reason is because the function that is used there.
Okay, I think we can move forward to Fabian. Will take us to AI in the context of chemistry and protein structure.
Yeah, you can see my presentation?
Yeah.
Okay, so in one comment only, maybe it's possible to avoid the notifications of the people entering and leaving the chat in the top. It will be, and if it's possible, I would be grateful. Okay, so you will see my left side because I'm looking at the big screen, so sorry for that. And Dvir and Roy made everything, made the work for me, because I'm going to talk about, not about deep learning. And the Nobel Prize in chemistry this year was awarded to Baker, Hassabis, and Jumper for two topics, actually, for protein structure prediction and computational protein design, which are related but slightly different topics. And first, a little bit about the people. David Baker is a biochemist and computational biologist who really led many groundbreaking advancement in protein structure and protein design. Okay, the main software he uses is Rosetta software. I'm going to describe it shortly. And he's a member of the United States National Academy of Science and one of the 100 most influential people in the health in 224, according to Time magazine. Demis Hassabis, Sir Demis Hassabis, actually, is a British neuroscientist, AI researcher. He's a really co-founder of DeepMind, which is a company later acquired by Google, which is a company that developed AlphaFold. And Hassabis is really an expert in AI modeling, and he created advanced AI models specifically for protein structure prediction. And this was the real thing that advanced the field very, very quickly and very fast into AlphaFold with unprecedented accuracy. And John Jumper is a chemist, an American chemist and computer scientist also. So those people are also computer scientists and chemists and physics also, experts in physics, and also work in Google DeepMind. And they also was named by Nature as one of the people who matter in science in 2021. So really, their collaboration is very, very critical for the success of AlphaFold. So I'm going to start by saying or by explaining a little bit about why they won this prize. So in 2020, Hassabis and Jumper presented AlphaFold, which was followed quickly by Rosetta Fold in 2021. And these programs really achieved amazing breakthroughs, several breakthroughs in different disciplines, which enable the accurate prediction of more than 200 million known protein structures in nature. And this made a profound impact in life science, as you know, accelerating research in biology, medicine, etc. Baker also is a did a lot of research in de novo protein engineering and custom designed protein, basically making new folds. And this also has a lot of potential and for drug discovery and synthetic biology, as you can create new proteins with new functions. So these two topics are really exciting and transformative, and that's the reason they got the Nobel Prize. So a little bit about the background of the importance and how we got to AlphaFold. So as we know, sequence-defined structure, okay, like Humphreyson stated in 1973, each sequence basically falls into a specific and unique protein, specifically and unique to its structure, which is due to attraction or repulsion forces. And protein structures are critical to understand function. So the more we understand about structure, the more information we have, the more we can understand biochemistry and protein structure in general. And the other important statement comes from Levinthal's paradox, also in the 70s, which states that the total number of possible protein conformational space is astronomical. I mean, even for a short protein, we have almost infinite different possible conformations, which obviously the protein cannot sample. And he asked, okay, so how proteins find the native fold? Okay, so there were two ways of thinking about trying to get information about protein structure. One by just getting the three-dimensional structure directly, and the other one by thinking about how we can predict the path to the fold. Okay, so why do we need predictions? Because you could think that we could use experimental methods like X-rays, but they are slow, expensive, and not always they succeed. So with the time, a huge gap between the number of sequences in nature, which is about 200 million today known in nature, versus the number of solved structures, 200,000, a little bit more, appear. And that's a problem, because we want to know, if possible, all the 200 million structures, but we know a little fraction of it. So quickly the question arises, can we predict protein structures given only its amino acid sequence? That's the problem. That's the problem people wanted to solve for 70 years. And now I'd like to state about the two different problems that they tackle. One is the structure prediction, which is basically finding the lowest energy structure given a sequence. So given a sequence, I want to predict the structure. This is protein prediction, structure prediction, or protein modeling. And the other opposite problem is having a structure. Let's find the sequence that stabilizes it. Okay, find the lowest. In other words, protein design is finding the lowest energy sequence for a fixed structure. So they are related but opposite problems. Okay. So at the beginning, the most straightforward approach for protein structure prediction was template-based modeling. Why? Because it's easy. It uses known determined structures of homologous proteins as templates to build structures. So this is a very good method because we know that structure is conserved. So even far related sequences, at the end, they have similar structures. But this works only if we have a solved three-dimensional structure. Okay, so this fails to complete three-dimensional information. Another method, another approach is by biophysical simulations. And this is the Nobel Prize in 2013. This aims to understand the process of folding or simulate the process of folding based on energy and equation of motions. But this works for small proteins, but is basically prohibitive computational cost. So it's not scalable, and we cannot use it for all the proteins, so we are limited also here. So people wanted a method, and that's what we wanted from the beginning, a ab initio template-free modeling, some method that is able to model protein structures without significant similarity to any known sequence, solved experimentally. So obviously, this is the most difficult type of predictions, and it took 50 years, but this problem was recently solved by AlphaFold and RosettaFold, which are similar software, by using deep learning, by using these wonderful methods that were just described by Dvir and Rui. So I will try to describe the main breakthroughs made by these people in order to reach the final solution. There are too many to explain all of them, but I will try to overview them at least. So structure prediction started at the moment we had the first solved experimental structure, which was in 1958. Kenro and Perutz solved the structure of myoglobin, and almost immediately people started to think, okay, I have already one structure. Let's try to imitate this structure by taking similar sequences and see if they fold in a similar way. And as the protein data bank grows, the data bank is a database with all the known experimental structures, there were enormous amount of different methods trying to do exactly that, take structural information, sequence information, and somehow predict the structure for sequences we don't have the experimental solution. And those were from different types. Again, we had homology modelings and threadings and ab initio methods, but all of them had very limited success. until the appearance of AlphaFold and AlphaFold 2 and AlphaFold 3, and other similar methods, which again uses deep learning. On the way, many different methodologies were developed by David Baker, starting from Rosetta, the first ab initio prediction method, and during a long time, from the 1990s until today, there were successive improvements of the methods in order to get to the final solution, as I said. So I will try to describe the different methodologies. So one important point, one important thing that happened in 1994 is that a blind challenge to assess protein modeling was established, in which research groups are required to predict structures from protein sequences for targets with experimentally known but not released structures. So they could measure how good the methods of protein structure prediction were, similar to the competition Dvir mentioned. So this graph is showing the advance of the quality of the predicted methods. Here we have a measure called global distance test, which is a measure of how good the model matches the experimental structure. Remember that they don't know in advance how the experimental structure looks like. So this is after the experimental structures are released. And starting from CASP1, this blue deep line, we have different GDTs for different, there are many different targets on each competition. We see here just the average line. And the interesting thing is that in the competition, they gave easy targets. We have a lot of homology to known targets, so they are easily done by template-based modeling. And difficult targets, which are those targets without any detectable homology, not sequential and not structural. And therefore, those are the really interesting targets, okay, that people try to different, I think, hundreds of groups submitting results to the CASP, and then they are evaluated and see which are the best ones. And actually, a GDT of about 90 means that the model is almost as good as experimental structure, because also experimental structure has, of course, errors. So again, this shows the advance, I mean, from 1994 to AlphaFold 2 in red. And I will show you exactly here, the AlphaFold 2 in 2020, basically the average of the GDT for AlphaFold 2 is basically 85 or 90. So they basically solved the problem. So they are as good almost for ab initio modeling, this modeling that doesn't use any template, that for template-based modeling. But this was a slow process, very slow process. Okay. And this is an example of the CASP14, a couple of targets that were solved by AlphaFold 2. And as you can see, the experimental result in green is almost exactly equivalent to the computational prediction in blue. So really the jump from 2016 to 2020 was really amazing. I mean, people were really amazed, and it was a quantitative and qualitative jump that allowed to predict ab initio structures. Okay, so what made this possible? Okay, this is a combination of basic research, big data, PDB, UniProt data, sequence data, structural data, and machine learning. Machine learning and deep learning algorithms, as so well described by Dvir and E. We had the power of learning and recognizing patterns. We had very high precision. In our case, recognizing patterns that connect somehow primary sequence and three-dimensional forms. And this is what I'm trying to explain now. So this is really the integration of this information that made AlphaFold breakthrough possible. So how this happened? And now here comes the different breakthroughs done by David Baker, several of them done by David Baker. There are other people, of course. So the first ab initio method that worked had the idea of using short sequence segments, adopt local structures similar to PDB fragments. So in other words, if I can identify short sequence segments of my target protein, for which I don't know the structure, which have structural information of similar sequences for these short fragments, I can try to imitate the structure of these short sequences for which I have the solution, and then I can try to assemble all those fragments for which I have partial faults. Okay, so the fragments are then assembled by Monte Carlo search in randomly combining these fragments. Okay, so in a sense, it's like making a Lego or assemble a Lego of different parts for which I know the structures. Okay, it's a Lego ab initio method. It worked quite well for small proteins. And then we need to refine the global energy. This is done by Rosetta energy function. This is basically pure physics and knowledge-based potential. But the idea is a fragment sample. Okay, this is the Baker's genius idea. And then I can show you how Baker presents the pipeline for this first initial Rosetta fragment program in 1997. So the first part, this is taken for his presentations. The first part is to identify local sequences. This is done by making a multiple alignment and then building profiles. And then we need to search fragments using this multiple alignment locally. And then we need to assemble. Okay, this is exactly what is going on. And the assembly is done by physics and energetics. This is a movie showing how one protein is assembled using Rosetta fragments. You can see in the movie that fragments are moving. I mean, they are not really, it's not really allowed to move specific resins. That's a drawback, but it worked relatively well for small proteins, well-folded globular proteins. Okay, you can see here again how it works. So this methodology is good for small molecules, but limited because it's very, very slow and computational demanding. So it's not very effective, and also limited to small and multiple alignments. The next step, the next breakthrough is also from Baker, in 20 years later almost, which is to use coevolutionary pattern. Already in the 60s, it was observed that co-evolving residues tend to be physically proximal. So if I build an alignment and I find in different species that there are compensatory mutations among these species, I can assume that in the three-dimensional structure, those two amino acids which are co-evolving will be close in the three-dimensional space. And this is very, very strong. This is known for a long time, but there were no enough computer algorithms, or not good enough computer algorithms, or effective enough in order to extract this information for the whole protein. Because imagine we have thousands of homologs in each alignment, and the protein can be very large, etc., etc. So the information is there, but it's difficult to extract. And here again comes the algorithms of deep learning. So now we will have the updated pipeline will be to build an alignment and then extract the co-evolving information using a probabilistic model to detect coevolutionary pattern. Okay, that is what Baker added to the previous pipeline of fragments. And from this detection of coevolutionary pattern, I could, in principle, build a residue distance map. So I can know approximately what will be the distance between each pair of residues in a specific protein. And that allows me to build a protein prediction or protein structure, protein model by restraining contacts, because I know which residue needs to be close to which residue, and then from here, use again the fragments. So here the pipeline will be updated, still the MSA, still the fragments, and now they added, they did an update of Rosetta, they call it Rosetta Gremlins, which extracted coevolutionary information and added that as a restraint with a contact map to the modeling process. And the modeling process is similar as it was before, but more effective and more accurate because we have more information about the context. Rosetta Gremlin coevolution improves ab initio prediction of the previous fragment I showed you, pure fragment method, but it's still too expensive to run routinely. I mean, 2 weeks with, I think, 1,000 computers per prediction. So it's still very costly. Here we can see a prediction versus a native. It works pretty good, not perfectly. We are still far from a GDT of 90 here, but it works. Yep. So the breakthrough number 3, and the most recent breakthrough, was done by Hassabis and Jumper. Here they come, they enter into the game with AlphaFold 1 and AlphaFold 2, which is, as described before, using deep learning in protein folding. Why? Because it's very effective. It's a very good method that is able to find very little pieces of information from very large data, as exactly as I explained before. So now the pipeline remains the same. We still need the multiple alignment, still need some method for coevolutionary detection, map, and structure prediction. But now the method to extract the coevolutionary information is deep learning. It's a model of deep learning, connecting the input and the output layer. In this case, the input layer is a multiple alignment, the output layer is a distance two-dimensional contact map with many millions of parameters. And this works exactly as described before. And it works because machine learning is very, very good. Deep learning especially is very good at pattern recognition that works by finding and focusing on the most relevant information in multiple alignments. So with the multiple alignment of tens of thousands of homologs and a very, very long protein, still is able to find little pieces of information about coevolution that they are translated into very, very accurate maps or distant maps, and then used for structure prediction. So it's really a game changer. The deep neural networks implemented in AlphaFold is really a game changer, and it was done by Hassabis and Jumper. That's the reason they got the Nobel Prize, their part. Since AlphaFold now is able to extract very accurate information from the contact maps, and then the building of the model is more accurate. And it's very fast, also very important, it's very, very fast. So we came from 2 or 3 weeks per protein to 2 or 3 hours per protein. So that's really, really very important in order to be able to predict many proteins. So AlphaFold is released in 2018. And the update here is instead of using the probabilistic method for extracting coevolutionary information, Jumper and Hassabis implement a ResNet deep neural net model with many, many hidden layers and billions of parameters, basically producing a very, very accurate distance matrix. So this is already a big jump, and the GDT average here is 60 something. It's not perfect, but it's very, very good and much better than before. It's a big jump, a big, big, big jump in accuracy. And then we build, after we have the distances, we build again the model by minimizing physics-based energy potential. So still the building is using something similar to Rosetta. That's the reason Rosetta is so important here. So the ideas were there, but the technology of the deep learning really improved that very much. And then the next jump, the breakthrough 4, I call it, is putting everything together. I mean, AlphaFold now will use neural networks for everything. Given an alignment, it will build the first network that will extract information from this alignment, build the histogram, and then the Jumper and Hassabis uses a second deep neural net in order to build the structure. As this is difficult to explain, but in a few words, this structure module basically consider the protein as a residue gas. I mean, we don't have here a physics-based nothing. I mean, there's no energy, no fragments, no protein, actually. We have little dots in the space, which are called residue gas model, which are moved by the network to satisfy distances and geometric constraints, which are learned from the PDB. So this network learns from the PDB, and the PDB includes enough information about the geometry and distances, etc., in order to be able to build the final model. This is even more effective than the previous AlphaFold 1. They also add additional things that improve the prediction, like refinement. This was also mentioned by Dvir. They draw the feedback loop of the 2 different networks. They talk between them and improve each other. So the distance matrix is improved, and then the structure is improved, and then the model is improved. Everything is improved iteratively. And they also use here, if possible, templates when available. And they also give an internal local accuracy measure, which is called PLDDT, which allows to see or to measure how good or bad AlphaFold thinks the final models are. This is very, very important for researchers to know if they can trust or not the models. This is really, really critical. So AlphaFold now is very entirely based on pattern recognition and deep learning. And the success is basically due to the superb engineering. I mean, the ideas were there, but they glue them together using deep learning. And today, AlphaFold is still the most accurate ab initio protein structure prediction method, which is very fast also. Again, this is very important. And again, I want to stress that the method doesn't reflect nothing about the protein folding. We don't learn about the protein folding with AlphaFold. We learn only about the final structure, okay? Something close to a native structure, because here we can see a movie of what AlphaFold is doing. So you see that during the process of constructing the model, very large non-physical distances, for example, are obtained. I mean, AlphaFold is not teaching us anything about the process of folding. And as you know, this has had a dramatic impact in life science. AlphaFold, for example, before AlphaFold, we had information about 25% of different proteomes, for example, Homo sapiens. If we put together the PDB information, experimental information, and homology models done with very good models, but when we add here the information obtained by accurate models obtained by AlphaFold, this almost doubles the accurate structural information we have now. So this is really amazing, and as you know, has a really, really dramatic impact. Another example is how good AlphaFold is that docking now can be done on AlphaFold models as good as on experimental structures, or very close to it. Here we have an example of an agonist docked onto the model and cryo-EM structure in cyan of the serotonin receptor, and they basically gave the same result. So really dramatic impact. Now, a few minutes I want to devote now to protein design. Protein design, as we said before, is the inverse problem.
Yeah,
okay?
I'm
yes.
jumping because I'm thinking maybe we should stick to time, and we are already.
Okay.
Maybe we should skip the design part. If you can jump to conclusion, I think maybe it's better.
Yes, sure. Yeah, okay. Let me go to conclusions. Yeah. Next time. So, let me 1 second. Here. So I just want to mention that this is only the beginning. I mean, there are now coming many models and applications that predict protein complexes of RNA, of DNA, using, for example, diffusion models, denoising models, etc., or even we can dock small molecules now to growing proteins around it, and then we can do docking even for very deep pockets, etc., So this is really the beginning of a very exciting time of protein prediction. That's it. Thank you very much. And sorry for the timing.
Thanks, Fabian. That has been fascinating and very clear. And actually pretty much on time. I think we are still good.
Okay.
Questions. So
sorry for that.
You type in questions in the chat, that would be great. Or if you want to open your mic and speak, that's also fine. I can read some, I can read some questions that already are popping up. One is, what is the actual practice for being solved with AlphaFold? Are they put back to the PDB? And if so,
again, please.
And if so, is that a problem?
Can you please repeat the question? I didn't hear again.
What's the current practice for new structures solved by AlphaFold in terms of databasing? Are they put back to the database?
Okay. See, so if the models are stored again in the protein data bank, that's maybe the question. I mean, if that is the question, yes, the protein data bank now added about 1 million models by AlphaFold. So even the PDB, which is a very strict database which evaluates the models by stereochemists and distance and many other criteria, are trusting those models, if this is the question, if you understood the question. So the models are still models, we need to be careful, but they are routinely used for many things, as I said, studying drug design or drug delivery or docking or many other things. I mean, yes, they are very accurate. And yeah.
Thanks. I think the question is, given that that's the case, is that a problem? I guess, is that a problem from the point of view of the model learning from itself?
But you mean is a problem for the experimentalist?
I'm reading out the question. I don't know if Sarah, who wrote the question, want to clarify. I'm guessing, is that a problem that the model is starting to learn from its own predictions?
I understand now. No, because as mentioned before, AlphaFold is even able to produce protein hallucinations. I mean, the network learns from the PDB, but is able to predict things that it never saw, if this is the question. I mean, this is exactly what deep learning does. I mean, it's amazing. It's difficult to believe, but given even sequences that are not unnatural sequences, in some cases, the network and AlphaFold is able to produce a fault. And then these faults have been tested experimentally, and they indeed fall in the way AlphaFold predicts. Something never seen. If this is maybe a better answer to the question.
I think Sarah just clarified the question similar to how we framed it. I think we are fine there. Another question that appears is, can RF diffusion AA predict antigen for a known antibody?
Yes, they are working exactly on that. This is difficult topic because the antigens and antibodies, as known, they don't co-evolve. So if we don't have a strong co-evolution, this is difficult to predict the interaction, for example. But the RF diffusion, since they are based on more molecular geometry and probabilities of distribution of distances, they are better. They're not perfect, but they are better for antigen-antibody problems. This is a very good point. Yeah.
Maybe I can add a question.
Yes, of course.
Since the information of the evolutionary co-evolving residue is still important in protein prediction. Like if I have a protein that have just one sequence, no evolutionary homologs, how well can we predict?
Okay, there are models that try to do exactly what you mentioned. There are models which are trying to work without homologs. They still may work due to the way that the ASM fold, for example, works. They work like ChatGPT, just for trying to complete the structure in a way. But they work worse, but they are able to predict structures starting only for one sequence because in a sense you are trying to imitate what you have at the PDB. So if you have enough parameters, they tend to work. Okay, not as good as multiple alignment, but they tend to work.
Okay, I think we can conclude this part and move on to the last talk.
Very quick question to Fabian, maybe.
Okay.
Can I ask?
Sure.
Are the structures that have been predicted correspond to indeed to the minimal energy of these proteins?
Yes, they are very close to the minimal energy. As what we're predicting are things that are close to native. Okay, and even after AlphaFold ends its prediction, there is a processing that minimizes the energy. So it doesn't end there. They are not exactly in the minima, but they are then minimized by Ember or by any other molecular dynamics minimizer, and we get close to that. Okay, and there are also other types of predictions now that try to make noise on purpose on the multiple alignments, and then you can get an ensemble of conformation, not only the minima, but many conformations close to the minima. So even they are trying to use AlphaFold as a to imitate molecular dynamics, let's say in this way. But yes, the answer is yes.
No, but the question is the other way. Are there many structures that have been predicted that are not the minimal energy of these proteins?
From what I know, not. I mean, they tend to be in the minima. Otherwise, they will not fall. I mean, at least for globular proteins. No, the answer is I think they are close to minimal energy. I think this is what AlphaFold is trying to predict, things that are similar to what we see at the PDB. Okay, which are in the minimal energy or close to that.
Okay.
Thank you very much.
It's been fascinating and fantastic, and we're moving on to last presentation on the Nobel Prize in Physiology and Medicine for microRNA, awarded to Victor Ambros and Gary Ruvkun, will be presented by Ayelet Lamm. Thank you, Ayelet.
Okay, no. Can you see the presentation?
Yeah.
Okay, great.
We are seeing your present.
Oh. Okay, so so this Nobel Prize was actually made the WOM community really glad because of two reasons. Okay, one is because it's been a while since the Nobel Prize was given for basic science. And the second part is because we didn't think that it will be given to microRNA because small RNAs were already, RNAi was already was given a Nobel Prize for Andy Fire and Craig Mello. So both Victor Ambros and Gary Ruvkun didn't think it will happen. Okay. So everything starts with the worm. Sorry, I don't know what's going on here. So this is C. elegans, a tiny worm, and we have a few labs in the faculty that works on these worms, and they are amazing. You can see that they are transparent, so we can see everything inside, and they're also very small, so we can grow many of them in just one plate and look at them under the microscope, also do genetics and so on. And everything started with Cindy Brenner. So Cindy Brenner, together with Robert Horwitz and John Sarston, got the Nobel Prize in 2002 for basically introducing C. elegans. So Cindy Brenner was the one that introduced C. elegans to the research community, to the academic community, and he supervised actually, worked with Bob Horwitz and John Sarston, and basically he also was mentored everyone in the WOM community that got a Nobel Prize, including Martin Schiffli that got Nobel Prize on GFP, that was also in C. elegans, and Andrew Fire that got Nobel Prize on RNAi, and everything started in his lab. So Bob Horwitz did this postdoc with Cindy Brenner and he found, he got a Nobel Prize for it, on apoptosis, how the apoptosis program itself, he found the genes, and also shown later on that this mechanism is really important also in human, in basically neuronal diseases. So, so one of the projects Cindy Brenner did was to identify mutations in worms And one of the things that he did was to look at heterochronic mutations, and he found LIN4, and LIN4, a mutation in LIN4 in this time. It was I think early in the 1980s. They didn't have the sequence or the clones, but they had mutations. And LIN4, if this is the wild type form, if you can see the wild type form, LIN4 actually skip the first stages of development of the worm, and in LIN4 you get a worm that has a bag of worm, a bag of worm phenotype, which means that the eggs are inside the worm. This worm is pretty big. And later on, so heterochronic genes means that the mutation in these genes cause skipping or retention of some of the developmental stages. So these genes are basically very important for the development timing of the worm. So Cindy Brenner isolated LIN4, and in Robert Hovitz's lab, he isolated LIN14 as another heterochronic mutation. So LIN14 is actually really small, mutation LIN14, and it caused the worm to stay in early developmental stages, larval stages. Okay, so Victor Ambros and Gary Ruvkun, they both did postdoc with Bob Horwitz at MIT, and this is where they met, and they both work on LIN14. In order, so they did the entire postdoc on LIN14, basically to just find the gene. So they did RFLP, chromosome walking, and they published several papers. This is pretty funny because these are methods that we don't longer teach because we don't use them anymore. But they did the entire postdoc on LIN14 and heterochronic genes. And when they published several papers, the main paper on LIN14 was actually published by both of them in Genetics. And after they started their own lab, both in Harvard, they both continued to study LIN14 and LIN4 genes to understand better how the heterochronic genes works. So also in Bob Horwitz's lab, they found that LIN4 negatively regulate LIN14, and you can, as you can see from the phenotypes, if we go back, LIN4 is pretty long, and LIN14 is pretty short. Okay, so these are the three papers that they publish in their own labs, and that actually identified LIN4 as a small RNA that regulates LIN14. So these two papers were published in Cell in 1993, and these were back-to-back because they actually worked together in order to, they had their own findings, and then they talked to each other and publish the papers back-to-back. But this was published in 1993. They identified LIN4 as a small RNA. However, okay, they published it. It was published in Cell, but it wasn't very interesting. Why? Because the community thought, okay, this is probably a specific finding for C. elegans. It's not really interesting. But in 2000, Gary Ruvkun identified LET7 microRNA, and this is a very small paper that was published in Nature. And the reason is that LET7, which is also a heterochronic gene, identified in C. elegans, is highly conserved, and he found that LET7 is also in human. And this is where microRNA actually started to get attention. So the first paper that was published, and this was back-to-back with between Victor Ambros and Gary Ruvkun, and this came from Victor Ambros' lab. Just to note that the first author is Rosalind Lee. So there was a huge talk about how come Rosalind, which is also part of many papers from Victor Ambros, was not part of the Nobel. And Rosalind is actually Victor's wife, and she was there. They were married during his PhD, so it was before he has actually even started his postdoc with Bob Horwitz. But I think part of it is because of a name, which maybe said something about another Rosalind that didn't get the Nobel Prize. But in this case, Rosalind Lee was actually his research assistant. She was part of his lab for many years, including now. Okay, so in this paper, Victor Ambros actually worked more about Lin4 than on Lin14. So he cloned Lin4 by using, again, autoblot with radioactive and RFLP. And what he found was that if we look at the northern blot of Lin4, after many attempts of looking for a Lin4 sequence, he found that Lin4 is pretty short. From this northern blot, you can see that in wild type and in the rescue strain. So after he injected the cloning of Lin4. So this was published in 1993. Before many things that we use now, but the injection system, in order to get transgene into C. elegans, was actually constructed by Craig Mello and Andrew Fire, who actually got the Nobel Prize on RNAi. But the paper was published later on. But one of the big thing that they actually did was generating all the system for injecting DNA into the worm in order to make transgenes, and this is what they used. When they ran the northern blot, they found that Lin4 is actually really small. There is a short band around 22 nucleotides. And there's also a longer band, but only for 60 nucleotide, Lin4L long, but it's not very common, and they mainly see it in the rescue strain, probably because in the rescue strain Lin4 is overexpressed, so this is why they see it, but not in the wild type. And when they look at the sequence, and this was done again with Gary Ruvkun's lab, they found that they see a complementary between Lin4 and Lin14 triple UTR. So this part was actually done together with Gary Ruvkun's lab, which was interesting to them because they found that there is a short RNA that seems to complementary to the 3' UTR of Lin14, and it probably negatively regulates it. So in the second paper, this was with Gary Ruvkun, they looked at Lin14, and this is from the paper, and what they took was, they took worms that have a transgene, and they stained them with LAC-Z. So this is the blue. So the top are just worms. This is L1 larva stage. So this is one of the younger larva stages, and this is L4, the last larva stage. And they took a clone with LAC-Z and another gene, the ANC54, 3' UTR. And they use LAC-Z stain. So in wild type, they see ANC54 as a muscle gene, and they see staining in the muscles in both stages. When they took Lin14, 3' UTR, they saw that in L1 they see expression. However, there's no expression of Lin14 in the L4 stage. But when they took Lin14 with Lin14 3' UTR with LAC-Z with Lin4 backward mutation, they see that actually Lin14, the construct, is expressed in both the L1 and L4. So it seems that Lin4 negatively regulates Lin14, but in specific stages. And as they found before in Bobovich's lab, this is a heterochronic gene. Other findings from this paper was that, not just the temporal regulation of the 3' UTR, so that basically the regulation is only on the protein level and not on the RNA level. So it seems like this small RNA, Lin4, actually regulates Lin14 in the translation life of the RNA, and probably not causing degradation. And also what is really important is that it's not the protein sequence itself, but it's the 3' UTR who is very important. So these two papers were the foundation of identifying the microRNAs and how they work. And again, in 2000, Gary Ruvkun actually published another microRNA, Let7, which is also a heterochronic regulation. It also complementary to its target gene, Lin41, at its 3' UTR. But what is very important from this paper is that it's highly conserved, and he could identify Let7 in human. And also Lin41, which is the target in C. elegans, is also conserved in other organisms. So this started the identification of microRNA, how they work, where they are. And I just want to note that that although the processes of RNAi, which got a Nobel Prize in 2006, seems like it's really similar to microRNA. These are two completely different processes, although some of the mechanisms are identical. RNAi is meant for protection against viruses, foreign RNA. It binds double-stranded RNA and degrades it mainly. And in contrast to microRNA, it's not that conserved as microRNA. So RNAi the main mechanism that we know now is actually in C. elegans in plants, but it's very different in human. While microRNA is very similar in all the organisms it was identified in. And microRNA are actually genes. And since then, we found around 1,000 microRNA in humans. You can find them in Mirbase. They're all annotated there. And the idea with microRNA is that are basically genes that go to transcription in many cases. They can be generated from introns, from genes, but they are undergo transcriptions, usually by RNA pol2. They go through processing in the nuclear. After generating, so the transcription generates 3 microRNA and they have poly A. And after processing with dosha complex, they are to generate 3 microRNA, they are exported into the cytoplasm, and then they are processed by Geisler to generate the mature microRNA. And then by using the RISC complex and argonauts, they bind to the targeted mRNA, and they can cause degradation by deadenylation or in other processes, or attenuating the translation and basically reducing production of proteins. MicroRNAs were, so this is the main, what we think today, how microRNA are processed. However, it's getting to be really, really big microRNA because they are transcribed. They can be transcribed in specific cells. So some of them targets specific mRNA in specific cells. So they were identified to be important mainly for development, but also for generating cancer and other diseases. And they are also highly controlled. They have their own promoters sometimes, but they also, the expression is also controlled by, for example, there's stoichiometry. So, for example, so if you have circular RNA that can take all the microRNAs that their targets are, the targets are free to be activated or generate proteins. So there, the regulation is highly important for our life, basically. And because of their small nature, they're also extremely, extremely stable in their structure. So we can use them now for many processes for diagnostics and identification. But again, because of the small size and incomplete alignment with the triple MTR, it's really hard to identify their targets and basically identify them. One last note. So yeah, so here. So this was important. This is from the picture from the Nobel Committee. One last note is that microRNA, because of their, and also small RNA from C. elegans, because of the short nature, identifying them and cloning them was really a pain because it's really hard to just identify 22 bases. So part of the high-throughput sequencing protocols that we are used now was actually invented to identify small RNAs. And all the things that we use for for sequencing RNA. And all of the tweaks and changes in the promoters and so on, in the adapters, were actually invented to identify microRNAs. And this is actually started a lot of the, for example, strand-specific RNA sequencing, single-cell RNA sequencing, and so on, that was actually started from the BRAVE protocol. BRAVE because they were really difficult, that were invented from Victor Ambros' lab, David Bartel and Tushel, and for identification of microRNAs for RNA. So that's it. Sorry.
Thanks much, Ayelet. This has been so fascinating on all accounts, but mainly the kind of the putting it all in the correct historic context was very, very interesting. I'm trying to, there are no questions yet on the prompt, but maybe I can start. It wasn't clear for me. When was it established that it's general in the sense, not in the sense of a cross-phylogeny from worms to human, but in the sense that you can actually use it to shut down any gene of you would wish. So when was it? It was first demonstrated you can actually design microRNA for a specific gene and shut it down.
So the thing is that RNAi was actually published in 1998, and RNAi is a perfect match. So all of the techniques like shRNA that was used in human later on was basically were on the concept of RNAi and not on microRNA, because because to structure a specific microRNA that targets a 3' UTR, which only the 3D region of the microRNA, which is only 7 to 8 base pairs, these are the perfect matches for the 3' UTR, is really, really hard. And with the antisense and siRNA, this was actually the ones that went into the clinic and looked to silence genes. So microRNA was, this is why they didn't think they will get the Nobel Prize, because they identified genes that regulate other genes, but it wasn't really used in a clinical setup. So shRNA that came from RNAi was the one that mainly used. To engineer a specific microRNA for specific gene is really complicated. While shRNA is pretty simple with the antisense oligos that comes right now, is much better. So yeah, so this is why this is more basic science than basically shRNA.
Okay, the questions I'm reading out, it looks, I guess it looks like they were lucky. The first ever discovered mRNA fully matched its target. I'm not sure I understand the question. Maybe
yeah, they were lucky. they were looking for heterochronic genes, but yeah. But if you see the history, that's really amazing. Like there were a huge group in Cambridge that Cindy Brennan actually got everyone there, like Martin Schiffley with the GFP, Bob Horwitz, Salston, Andrew Fire, and everyone. All at the end got Nobel Prizes, and there are lots of others too, like Judith Kimball and others that every one of them actually did really, really basic science. They did RFLP with chromosome walking, northern blots, and things that I don't think that actually students know what to do right now. So this is amazing, like a huge, amazing history. And I don't know if people are actually doing it right now. So yeah, they were lucky, but there's a lot of luck there. Like they were really search for everything that they could find in C. elegans. Cindy Brenner did a huge databases of mutants. We still use them. Like every paper that we get out from my lab, and I think most of the labs, we have reference to Cindy Brenner's paper. So this is pretty amazing.
More questions. Is microRNA, I guess, does microRNA only work on the 3' UTR?
No, but mainly. So the problem is we don't know the targets. So the seed region is only 7 bases, and there are prediction tools that try to identify the targets from the microRNA. The one that most people use is target scan. They also identified targets not just in 3' UTR, but we don't know. And also there is a huge redundancy between the microRNAs. So if we look at the 3' UTR that is targeted by microRNA, we can see several positions of probably microRNA that targeting this specific 3' UTR. And also the microRNAs themselves comes as a group, and they might be different in just one nucleotide. So it's really complicated. And because we don't know exactly what the targets are, we don't know exactly if this is just 3' UTR specific or not. And also circular RNA don't come from the 3' UTR, and we know that they also target microRNA.
Another question, people are asking, how strong, for genes that are regulated by microRNA, how strong, tight is that regulation if you compare it to regulating the transcription or regulating protein stability, degradation?
It varies. We know that mutations in microRNA can cause basically lethality in human. So the regulation is really strong. But it varies, depends. So again, the process is really complicated. It changes from a cell to a cell. In some cells, you see like middle regulation. Some is generated, some is not. In some cells complete regulation, in some cells none. So it varies. I can't tell specifically on like globally on the entire regulation.
Okay, I guess we can stop here. I want to say thank you to everyone who participated. There seemed to be like around 100 people, so I think it seemed like it was timely. And I want to say thank you to our speakers, to Dvir, to Fabian, to Ayelet. It's really, I mean, took this challenge very quickly, and within a couple of weeks put together brilliant, fascinating talks. So thank you for coming up to the challenge. Much thanks again to Sivan Geisler-Edelbaum. And Sivan, it seems like you have worked to organize the same thing next year. So thank you, everyone. It has been
Thank
fun.
you very much.
Enjoy the rest of the day. Bye-bye.
Yeah, you too. Take care.
Thank you.